Everybody, and welcome to our third Builders Group talk series today. Again, thank you for joining us here on Zoom. My name is Jessica Lim, and Gail Bob and I are the co-chair to BINA's Builders Group. So for today's talk, we will first start with 15 minutes presentation given by each speakers, and we will end the workshop with a 30 minutes interactive Q&A session. So if anyone has any questions for any of the speakers, please feel free to put it in the chat box or unmute yourself to ask questions. So we will first start with our first speaker, Kevin Dean. Kevin, if you want to pull up your slides. Kevin is an assistant professor at Lyda Hill Department of Bioinformatics at UT Southwestern Medical Center. His lab focuses on developing self-driving super-resolution microscopy to help identify metastatic colonization events in large tissue volumes. And today he will be presenting his team's development on smart light sheet microscope system. So Kevin Dean.
All right, thank you so much. It's great to see so many familiar faces and names in the chat. Today I'll be talking to you about our dissemination efforts, which really kind of fits into this open hardware build session quite nicely. This is part of a NIH-funded effort, the so-called RM1 or BTOD centers, the Biomedical Technology Optimization and Development Center. So great opportunity for funding through the NIGMS if you have a sort of technology that you want to kind of find mechanisms to disseminate. And so this audience is probably pretty familiar with the joys of aligning optics by hand. It can be very zen. It can also be very frustrating. So the vast majority of systems that we first started out building really use these free space optical systems where you literally placed and shimmed every single optical element on the table, measuring back reflections or focus position or collimation, and doing this step by step by step until you got to the very end, and it could take a long time. And so the vast majority of labs out there aren't going to take that approach. And some of these methods that actually reduce the degrees of freedom slightly also don't necessarily provide exactly the solution that you want. So anyone that's probably used a cage system has had something aligned just right and then timed it and secured it and then see the beam deviate. That's not the most inspiring or faith-inspiring part of that technology. And so our goal was to really kind of translate from these classical free space cage or rail systems, and then to establish precision base plates that could be easily built by non-experts. The whole goal is for you to be able to have the confidence. This may not be the audience. This audience has the confidence to do it. But what would it take to actually get a biologist without any optics experience to build a system? And what would it take? And so our goal is to really reduce this alignment burden and also the operation burden. And so we've been trying to generate a strategy where we publish the entire transfer mechanism. So how do you go from a design all the way to a functional system? And that's includes the control of the system, the software layer, the firmware layer, the hardware layer, all the way down to the actual optical system. And we're trying to do this in a way that allows you to address different biological problems with different sorts of technologies. So the very first part is software. So every system that we have needs a unified control layer. And what we've done is created Navigate. If anyone ever wants to try this out, we're happy to help. We have dedicated people who can help you get it up and going. My hope is it shouldn't be that tough to get up and going. But the whole idea is we can now automate complex imaging workflows. You can do event-driven microscopy so that you can change the way the system operates, maybe go from a fast imaging mode to a slow imaging mode or vice versa, or have a macro scale view, identify a feature of interest, zoom in. Or you could even have things like adaptive optics where you're now running an iterative routine and adjusting the wavefront to see if you can optimize the performance of a microscope. It's all very reprogrammable or programmable, and you can do this with a GUI interface so that those who may not feel confident in their programming capability can literally just drag boxes around. And the hope is now we have a unified system that can now standardize protocols across instruments. So you might develop a smart imaging workflow, and then you can plug and play it directly into another microscope and leverage that. And all the data comes out with these new, block, like N5 ZAR, chunk-based, pyramidal-based image formats that really allow you to scale your image analysis. So the design process is quite repetitive, but we've tried to essentially emulate what, a professional, big microscope company will do. So we start from first principles and we design the system around the biological problem that we have. And then we go and say, all right, these are the lenses that we would need to achieve, this field of view, this sort of light sheet diameter, light sheet extent. And then we go into a optimization phase where we then use Zmax to optimize the position of every single element in the path. And that also includes a tolerancing analysis. So we can say, all right, if we mess up lens number 3 here by 0.05 degrees, what's the impact on the actual final output? And how does that compare to standard machining capabilities? And so we go from first principles to simulations, and then we translate those simulations as optimized beam positions or lens positions into actual physical coordinate spaces. So you can take the simulation and translate it into a mechanical post that, should we have the right base plate, you would know exactly where to position that optic. And so to kind of really even refine that further, we have the Polaris imaging systems from Thorlabs on all of our illumination units. So those have dowel pins that also restricts your rotational degrees of freedom. And so we then come and we have kind of a very simple illumination path shown here. And on the detection side, we've actually used predominantly ASI-based components so that you can now have just a single lens, your objective, your tube lens, your filter wheel, and your camera on a monolithic path that's also quite easy to assemble. So we have 3 different versions now. We started simple and are growing in complexity. And so we have the LSFM, or just standard kind of adherent light sheet or adherent cell imaging system. And that was published in eLife. We've moved and built an axially swept light sheet microscope version that provides imaging in different environments as well as different refractive indices. And more recently, in collaboration with Mehta, we actually put out a direct view oblique plane microscope as well. So the adherent cell imaging system, which we call Altair light sheet fluorescence microscope, essentially provides about 235 by 235 by 350 nanometer resolution on beads after deconvolution. This is on par with some of the most competitive light sheet microscopes in this imaging geometry, which makes sense because we're all working with the same objectives and we all deal with the same laws of physics. But we're able to see very nice details. So this is what the cell looks like as we actually acquire the entire image stack. Once we shear and rotate the data, we can see nice features, including, nucleoli. We can see individual microtubules, this nice actin cytoskeleton with these transverse arcs, as well as the small perinuclear Golgi. Now, the next system, we've moved up in complexity. So we have a single base plate, and it can now be configured in 3 different formats. And so we have the light sheet fluorescence microscope that I just presented. We now have kind of a truncated build where you can then, on the left, achieve the same performance. Where you have on the bottom and the center, and on the bottom right, you have our axially swept light sheet microscope and our clear tissue light sheet microscope. And what these do is essentially change 2 optical elements on the illumination path: the very last tube lens and its position, as well as the illumination objective. The ASLM uses the same detection objective that the adherent cell imaging system does, but the clear tissue also has a multi-immersion lens from ASI. So we've built one of these. the nice thing is, again, the simulation and the optics, the actual experiments match pretty well. We see that we predicted about a 392 nanometer beam waist at 488, and we get about 420 in our ASLM configuration. And we actually surpassed the ZMAX simulations at both 560 and 640. The final raw resolution here for about 170 micron field of view in both X and Y is about 320 nanometers in X and Y and about 350 nanometers in Z. So that's getting to be pretty good for imaging things like cells in reconstituted ECMs or extracellular matrices. So we just now really started to kind of benchmark this on biological experiments. So we imaged a zebrafish here, and we could see the formation of the vasculature and see this really nice, beautiful kind of migratory behavior that's initiated by cells kind of creating these like spiky filopodia. And this is inside of the fish. So the imaging is actually not incredibly easy, but overall, like quite pleased with those results. And so lastly is the oblique plane microscope. So oblique plane microscopy is incredibly useful. I mean, there's a lot of kind of dirty secrets about light sheet microscopy that include that if you're going to use water dipping lenses, it's going to be hard to keep your cells sterile for long periods of time. And there's also lots of different things that can drift, especially once you've got a heated chamber. So oblique plane microscopy provides a lot of the benefits of a regular inverted microscope. You can bring the objective up, you directly interface with the glass, you can have an autofocusing mechanism if you'd like. You can keep your sample sterile, you can do multi-position imaging. If you have large things, you can actually image, you can scan that thing in each dimension quite far. And so there's a lot of advantages. And so what we did with Reto's lab is actually design one using fast photography lenses. And the idea is that these lenses have external back pupil planes. And so we can essentially flip them in their opposite direction. And I can't really use my pointer, but you can actually then conjugate the pupil planes without having any additional relay optics. And we can actually have enough space to put a filter wheel and also a fold mirror between these two things. And so we've done this. We now have essentially a detection module, which I'm showing here, which consists of a focusing stage, this custom base plate, 2 photography lenses, a right angle mirror, a filter wheel, and a camera that's mounted on a rotation stage and translation stage. We then built an illumination unit to accompany it. And this thing is going to come in and launch the beam from the side. And that allows us to do something that's quite compact. We've added a fold mirror here to really make it as compact as possible. But ultimately, the beam comes up and is launched at that angle, which is kind of best seen here. So we put the specimen on top of a flat top stage. The entire detection system can go up and down and focus. We can scan in X and Y for large distances. And we can now achieve resolutions that are on the order of about 2.4 by 3.3 by 18 microns. And that's all done by projecting that image directly onto that camera. So we've done a couple prototype experiments. I'm pretty excited that you could actually see individual microtubules and expanded cells with a photography lens, which I think is pretty cool. And then we can also image things that are quite large, like an expanded liver specimen, which actually can span upwards of 100 millimeters after expansion. And so that's the strategy that we've put out. Essentially, we try to publish more than just a paper. We do exhaustive documentation, all put up on GitHub.io. We have all our CAD files, ZMAX files, parts list. We have archived data that's put up on Zenodo, and of course, we have our acquisition software. And so we're trying to do this for every build. And the hope is that if you're just an end user, you can take these parts and build it yourself. Or hopefully with an audience like this, that you might actually be inspired and take some of our ZMAX models and develop something new yourself. So with that, I'll wrap it up. John Hogg is the postdoc who led most of the dissemination effort, and I owe him a lot. He will be here for 2 more weeks before moving to the Bay Area to join E11 Bio, so I'm incredibly excited for him. And then I've got Ty and Muaz, who are big leaders on the OPM project, and Zach and Annie, who both provided incredible help developing Navigator acquisition software. I appreciate it.
Thanks, Kevin. So we can have our second speaker, Gail McConnell. Gail, if you want to pull up your presentation. So Gail is a professor of biophotonics in the Department of Physics at the University of Strathclyde. Her research involves the development of new optical instruments and technologies for cell and tissue imaging. And today she will be presenting her group's innovations on multi-immersion objective as well as 3D printed microscopy hardware. So Gail.
Cool, thank you very much. Thank you, Jessica, and the organisers for inviting me to present today. Yep, so I'll be giving an overview of some of the work that's going on in my lab at the moment. But first, a little bit of history. So we have this gigantic lens called the mesolens, and the raison d'etre for the mesolens was having a large volume of capture with subcellular resolution throughout. So it's the unusual combination of 4 times magnification and 0.47 Na. This is a diagram on the left-hand side of the screen here of a standard optical microscope with the objective lens, tube lens, eyepiece, and human eye is the photodetector at the top here. Here is the mesolens shown to scale next to it, and that's just the lens. That's not part of the microscope. So because we need this high numerical aperture, our individual lens elements are commensurately enormous. So it's roughly the same length and width as a human arm, and we need to design the objective lens and then build the microscope around it. So we've published a lot of different work now using the mesolens. We hope it's an established technology, and you can pick a video and hopefully watch it play and it's not too jittery. So for example, here what we have in the bottom part is the green contrast coming from human palatine tonsil, the magenta contrast that's just appeared coming from bacterial infection. And the rhetoric that and the dogma that we understood in human tonsillar diseases was that the bacteria resided solely in the tonsillar crypts, and our work has proven otherwise. So the mesolens has found utility in different areas of bioimaging, from looking at trypanosomes within intact living and fixed and cleared mouse brains, through to clinical applications, and in microbiology looking at macrocolony biofilms. The difficulty is that people would like a mesolens, and they're not easy to make. It takes about a year to make a single objective lens because we're using classical lens grinding, polishing and mounting techniques. So our answer to this so far has been to have a small imaging centre at the University of Strathclyde, which we call a mesolab, and people send us samples and we image them using a variety of different modes that we've developed for the mesolens, like bright field, dark field. We've got some phase contrast and point scanning methods, as well as some inbound multi-photon, which is quite new and exciting. We do some light sheet and also super resolution using computational methods. But because people understandably want this technology in their own lab or institute, we've turned our attention towards trying to democratise access to making the lenses themselves. Now, we're a long way from being able to make a mesolens using 3D print methods, but at least we have some of the groundwork now done that suggests that we can produce lens qualities that are suitable for optical imaging. So for this, we use off-the-shelf printers you can buy on Amazon or alternative vendors, and commercial resins. And when we use our clear, transparent resins, we end up with a preform that looks like this unprocessed lens in the top left-hand side. Following a simple spin coating step, this makes this rather diffuse-looking lens optically transparent, and we can get lens transparencies in upwards of 90% without any additional coatings. And they compare very favourably to the commercial off-the-shelf components. We get their STL files, thanks very much to the vendors, and then we use them for printing of our lenses. They have comparable curvature and optical performance to the commercial counterparts. What we do notice, however, is that the lenses that we can print are best matching their commercial counterpart when they are of short focal length, short or small radius of curvature, rather than the larger, flatter lenses. But there's things we can do about that. Just if proof were needed that we can use them for imaging quality work, what we have on the right-hand side here is a tissue section. So this is plant, it's a lime tree stem and is stained with osmium. So we have here, through a region of interest, line profile, resolution in the order of a few microns, but this is just taken with a single spherical lens element, so there's no post-processing correction. These are raw data. However, the real power in being able to make your own lenses is to make things that you cannot buy. So, as an example of this, what I'm showing here is a rather unusual bespoke element that we have designed and 3D printed, which we call an integrated lens coupled biprism. And the reason for that is because it is a lens coupled to a biprism. We have used this in a commercial Greenough style stereo microscope to improve the image contrast. So in the ordinary stereo microscope, the light source reaches a ground glass diffuser, the light is diffused onto the specimen plane, and then the light that is transmitted through the specimen is sent up each individual eye path towards the human eye. When we integrate the lens coupled biprism into the optical path, it performs two functions. The first is to act like a condenser lens for that incandescent source, and then also to steer the light in a more selective way along the optical path towards the eye as the detector. So you retain the stereo microscope view. The specimen here is onion epidermis, and it demonstrates it's been imaged with and without the lens coupled biprism to demonstrate the utility of this unusual optic. So on the left-hand side, we have a full field of view of our onion epidermis. This is a living sample, and then a region of interest showing higher magnification, but with no additional change in numerical aperture. With the lens coupled biprism on the right-hand side, even at the full field of view, when the static image, you can get a sense that there is an improved contrast. And then when we zoom into some of the smaller regions of interest, then we see that. But if I press play in a movie, hopefully it will. What we can see here in this movie, we do have that lens coupled biprism, is we are able to now see and resolve cytoplasmic streaming events, which are completely invisible in ordinary stereo microscope. So we are able to improve in this robust and used and accessible stereo microscope technology. At the other end of the spatial scale, what we have is a solid immersion lens, which we can make using very simple methods that don't even require a 3D printer. We take some of the 3D printing resin that's optically transparent. We deposit using a Gilson standard pipette, a 7 microliter drop onto the sample. We cure it using a torch or a UV laser pointer, as long as there's enough UV light there to cure the resin. It takes about 5 to 10 seconds. And in doing so, we can produce a glass hemisphere that's in the order of a few millimeters in diameter that costs less than 0.00005 cents. So it's a completely disposable part. And it's a great student project beyond anything else, but it does have application. The SIL improves the magnification by approximately the refractive index difference. So if the refractive index of the resin is 1.5, it should in theory increase the magnification by the same factor. We see closer to about 1.4 times magnification. But what we do see is a resolution improvement on the same scale. So our resolution goes from 550 to under 400 nanometers. Now this is a modest improvement, but for the amount of time and effort spent, it's actually quite beneficial. So here's the SIL applied to one of our samples. This is a section of mouse paw, and you can see through our region of interest here, as we apply across striated muscles, the striations are not visible without the SIL, and with the SIL, they are. We are also printing whole microscopes. So using the open flexure base, we have now a prescription and protocol for printing of an open Fourier tachographic microscope using commercial parts entirely, and 3D printing resins and printers that are widely available. All of the parts and code to run the microscope are freely available. There's a link to the paper here, which is currently in review. So on GitHub, everything is documented, and if anyone wants to try it, that would be great. But our reason for doing this is to, and I'll perhaps move past some of the results, you can see those in the preprint, is to address a particular concern in crop health, where we are currently facing a phosphate crisis. So phosphageddon, as I believe it's been called, has only been worsened by political events in the Middle East at the moment. And there is a growing concern that access to fertiliser, and phosphate in particular, is going to threaten our food security. And if we are required to grow our crops in low phosphate environments, they may be more susceptible to infection and disease. So we've performed a bunch of experiments growing plants in low and normal phosphate conditions with colonising pathogens. In our case, we're using Pseudomonas aeruginosa, and we've written Python-based code to measure and quantify the pathogenic burden in these data sets. Now, it's very rare that we have a good news story in science, but this is emerging as one, and it transpires that there appears to be no difference in the growth, in the colonisation behaviours of, admittedly, in these plants and these pathogens, irrespective of the phosphate conditions. So perhaps this means a slightly brighter future for our food and crop securities, but also a nice application for some of the technologies that we're developing. So with that, I'll finish up, acknowledging everyone in my lab and elsewhere that's been part of the work that we are doing, funders for making this work possible, and you for listening today. Thank you so much.
Thank you, Gail, for your presentation. Next up, our speaker will be Alfred. Alfred is a staff engineer at Calico Life Sciences. He specializes in applied physics, optics, mechanics, and instrument programming. He is widely known in the microscopy community as the inventor of the famous Snouty objective lens. Today, he will be presenting his team's developments on the single objective light sheet system. So, Alfred. Ooh, you might be muted.
Is that better?
Yes, there we go. We can hear you now.
Great, thanks. Yeah, so I'm Alfred, and I'm going to talk to you guys about the Snouty light sheet microscope. So let's just jump right in here. And I wanted to start briefly just with some motivation and a challenge, right? Why are we doing what we're doing? So for a really long time now, the confocal microscope has been like the go-to instrument for 3D biological imaging. And here, we build up the image point by point. And this process is both slow and delivers a high photo dose to the sample. So there's potentially a better way to do this, right? And it's called light sheet microscopy. And in this arrangement, we fire a sheet of light into the sample on the focal plane that we're looking at, and then we collect all of that fluorescence in parallel, right? So in principle, this is much faster and much gentler. Now, despite all of the advantages of light sheet microscopy, the reality is that the confocal microscope is actually still way more popular in practice. And there's a simple reason for this, and that's just that typical confocal microscopes respect this planar interface here that I'm showing with this red dotted line, where you have, biology on one side, optics on the other. And a traditional light sheet doesn't, with this orthogonal objective arrangement, you have the optics and the biology all mixed up together. So, regular confocal is compatible with slides, dishes, multi-well plates, microfluidic devices, and a traditional light sheet geometry isn't. So it's really that simple. So the challenge is, can we have an instrument that's kind of the best of both worlds, right? Can we have a light sheet geometry that's fast and gentle? And can we have it in a format that is convenient, right? That has this planar interface. Okay, so that's the challenge. How are we going to do this? what we want to do is take a regular light sheet where you have an objective that fires a sheet of light in from the side usually, and another objective that collects that emission light. And we want to somehow remove this optic here. So one way you could imagine doing that is taking a regular microscope that looks something like this and firing a sheet of light down through the microscope and tilting it over like I'm showing in this cartoon. Now, you can only tilt it so far, right, to this kind of maximum angle here, which is limited by the primary objective here. And then you can try to image this tilted plane down here. But there's a problem, which is that the light sheet actually isn't in the traditional focal plane of the objective. And so when you try and image this tilted image down here, what you get is something that is both distorted and highly aberrated. So whilst in principle, it's something that we would want to do, in practice, it really doesn't work very well at all. So the question is, how can we do this, right? How can we access this tilted plane here? So it turns out there's a clever way to do it. We can use something called remote refocus optics. And what we do here is we take our sort of conventional microscope and we add some extra optics downstream. And together, these optics provide a remote 3D copy of the sample, right? And this is very powerful because with this remote 3D copy, we can then use a special glass-tipped objective lens, which I'll call the Snouty objective, to image this tilted plane here at high resolution and losing very little light during this operation. So this is kind of a big deal, right? Because what we're saying is we can have, a light sheet microscope that is actually compatible with a standard microscope. So we're able to image this tilted plane here. We're able to put a light sheet on that tilted plane, and this is now compatible with all of your standard samples. Okay. So what I've shown is the ability to image a tilted plane, but I didn't say how we were going to get 3D data. And there's lots of different ways to do that. But a compelling way to do it is to use what I'll call a fast optical scanner. And what that does is it sweeps the light sheet back and forth in the sample to create this volumetric data. And again, this is kind of a big deal because the same operation with a confocal microscope gets you one line of one image. But here with the light sheet geometry, as we sweep the light sheet through, we actually get a whole volume for that same scan. Okay, so this enables routine imaging in 3D and effectively a new spatio-temporal regime for samples and for microscopy. Okay, so that's a lot of conceptual stuff, but what's the result? So basically what we have is a new class of microscope, which I'm showing here in this animation. We have this fast optical scanner, right? That sweeps the light sheet back and forth quickly in the sample. We're then able to collect all of the emission light down through this optical train. And if we go all the way back now to the Snouty objective, which sits down here in the remote space, this special glass tip lens is able to image that tilted plane with high efficiency and return almost all of that light to the camera. All right, so the first prototype looked something like this. And here I am driving around in 3D. Here's some cells on the screen. And every time the laser flashes up there, I'm taking a whole volume. So it really is a very fast technique. We'll just watch that laser flash a couple more times. There we go. So what does the data look like? Well, the first thing you notice when you use the light sheet is the resolution is really quite high for a light sheet. So, something like 300 nanometer XY. And you'll also notice, because it's a light sheet, that it's gentle and fast. So in this panel on the right here, you can see these natural killer cells chasing down a target. And this is volumetric imaging at a pretty high rate, and you can see that the cells are basically undisturbed by the imaging, which is fantastic. And just another couple of examples here. On the panel on the left, we're looking at yeast dividing over 12 to 14 hours. And they're just happily, going about their business. And they're really very small, 3 to 5 micron diameter, and we're getting this really high quality imaging performance here. And on the right-hand side, slightly larger, we can see some Jurkat cells dancing around happily. This is a multicolor volumetric time series. So after we developed this initial prototype small field of view objective, we then went on to develop a large field of view equivalent. And this was released in the form of the Dashi microscope, which extended the field of view towards the sort of millimeter scale. And you can see that here with this nice zebrafish embryo. Okay, so that covers kind of the base architecture, if you like. But what's cool about this platform is there's some really interesting upgrades that you can add to it. And one of them that I wanted to show is this any immersion concept. So what do I mean by that? Normally on a microscope, a regular microscope, if you want to perform 3D imaging, you'll do something like a Z-stack, right? You'll move the objective up and down and look at different focal planes. Now, at high NA or high resolution, this only really works well if you did a good job of matching your sample refractive index to the immersion of your objective. So in this case, I'm using a watery sample and a water immersion objective, and if you do that, it works really well. If you don't do that, for example, if you use an oil immersion objective, what you notice is the deeper you try and image, the blurrier it gets. And so actually, this doesn't work very well at all, especially at high numerical aperture. Now, you remember that the Snouty microscope actually uses these remote refocus optics. So a few years ago, I asked myself the question, well, what happens with remote refocus optics when we do, this sort of strongly mismatched immersion? So here I'm using an extreme example. I have an air objective, and I'm trying to look deep into a watery sample, right? Which is something that you would normally not try to do. So it turns out that if you configure this just right, where you put the traditional focal plane at what I'll call the depth 0 image plane, which is just at the surface of the sample or just above the cover slip. If you do that and you tune the remote refocus optics specifically for the refractive index of the sample, so if you do those two things, actually this works perfectly well. So this is kind of a big deal because what we're saying is that we can use an air objective, for example, to look deep into a liquid like water or an oil lens, or we can use like a regular water lens to look into a watery sample or any sort of variation or combination of these things. And there's different pros and cons to the different regimes. So if you use an air objective, then it's convenient. You could do things like high-speed tiling, or you can have like a thermal air gap. Maybe that's nice. If you use, say, an oil lens, you can get the maximum numerical aperture. And if you use a water lens, in this case with a watery sample, then you have like a boosted focal range because you have not just the remote refocus range, but you also have the standard focus range, and you can use them together. Okay. So I called this any immersion remote refocus or air microscopy. And how does this relate to the Snouty? Well, the original prototype was basically stuck with one particular choice of immersion type and one sample refractive index. But now we have this new any immersion version that I'm showing here. So this is cool, right? We have the air, the water, and the oil objective as options. We're using the large field of view optics in this design. And we can now look at some data and sort of see how this thing performs. So we'll start off with some mitochondria. And when you use this sort of new version of the microscope, the first thing you notice is like just how much larger the field of view is. It really makes a huge difference. And not only that, but here we're using the air objective to look into this watery sample, right? So normally wouldn't do that, right? And as you look deeper in Z, you'll notice like the resolution doesn't really seem to degrade, which is what we expect from theory, but it's kind of surprising just how well it works in practice. And we can kind of go crazy with this technique now. So here I'm showing an amoeba crawling around on a cover slip. This little red box here is the original range from the first prototype. But this scale bar here is 100 microns. So we're going like 120, 130 microns into this watery sample using a high NA air objective and getting this really nice diffraction limited performance throughout the volume. So really quite attractive. And the nice thing about the microscope is the flexibility, right? So if you want to go back, in this case, to the water objective and go deep, which is what I'm doing here to look at these neurons firing away, then you can do that. But I think really the air objective is the compelling mode. So here I'm showing like a sort of high-speed tiling of a 384 well plate in 4 colors. And you can just kind of whip around this thing without worrying about immersion and those effects. And the focus lock is really excellent because you have a strong reflection there to lock onto. So the data is crazy, right? So for this acquisition that I'm showing here, which is just one volume and one time point, it's almost a terabyte. So you can imagine if I did some kind of time series or tiled more fields, this would get out of hand pretty quickly. So I really think this is the future of high content imaging. We might just have to wait another 5 or 10 years before it's out there in the wild. If I've got time, I want to touch on one more mode that I think is interesting to upgrade this microscope, and it's a relatively easy upgrade. So we're going to take a fast microscope and we're going to make it even faster. So all the data I've been showing so far is 2D projections of 3D data, because it's convenient to show it that way. But we're taking 3D acquisitions each time. There is a way to run this microscope where you add an optical scanner at the end, just before the camera. And what that allows you to do is take 2D data directly. So what do I mean by that? Don't have time to really get into the details, but basically you can take fast 2D projections of 3D samples with one camera exposure. And you can do that in different directions or at different angles. So you can see that here with this cell that came from Redo's lab. It's a really nice example. And here, this is an example that I built myself that really kind of shows that ability to look at these different angles, which you can choose arbitrarily. And I'm taking these fast 2D projections. And the reason why this is so powerful is if you have a sample where you didn't need a full 3D volume, and actually just a couple of views was enough, and you want to run very quickly, then this mode allows you to do that. So here's the beating heart of a zebrafish with two, I think, orthogonal projections in this case. So, it takes a fast microscope and makes it potentially 10 or even 100 times faster. Okay, so that's all I have time for now. But I just want to say, this project has had really great adoption. There's at least 25 of these systems in the public. And I always like playing this. It's kind of my favorite animation from the project about how we make the Snouty objectives, which was volunteered by Lockie, I think 5 or 6 years ago now. It was like on Twitter or something, but I just think it's funny, so I'm going to show it. And of course, the most important slide perhaps is just to acknowledge everybody who contributed to this project, which is just so many people now that it's hard to really go through them all. But it's been a great effort from the open source community. So yeah, I'll stop there and we can chat later in the questions.
Great. Thank you, Alfred. Great talk. And then now we will have our fourth speaker, Shalin Mehta. Shalin leads the computational microscopy platform over at CZI Biohub. His team of machine learning and R&D engineers integrate optics and machine learning to map and model dynamics cellular systems. And today he will be presenting his team's latest work on high throughput label-free cell profiling. So Shalin.
Thank you, Jessica. Thanks, Kevin and Gail. Great talks. I'm going to talk about computational ideas. So this is slightly different from open hardware concepts. This is really about open source software that complements simple hardware and makes it useful. So maybe I should recap where I am. The new organization is now called Biohub in October last year. All the different sites that were supported by CZI, they unified their efforts, and now we are a single entity called Biohub. The Biohub sites that I'm part of, they are in Redwood Shores and Mission Bay. Those two sites are now a single organization. It's Imaging Grand Challenge. And the focus of the whole organization now is to pursue team science and to drive towards new methods for n-dimensional imaging across different biological scales. The site, Redwood Shore site, focuses on using electrons to look at molecular complexes. At San Francisco, we have a focus on using different types of light-matter interactions to map subcellular dynamics and organismal dynamics. And the goal is not just to visualize the biology, but it's also to train predictive models and put them in line with imaging and eventually perturb biology, do optical perturbations and other types of perturbations, molecular perturbations. So that's the organization. We are now reimagining a lot of our work. And as part of that, my team's effort has shifted from building microscopes to building computational frameworks that can be used to do scalable profiling of biological systems. So I'd phrase that problem as through this diagram. In the end, we want to look at some soft matter and read out biological activity. We want to read out some molecular responses. We want to read out what happens to organelles, how they interact, and read out what happens to cells in a tissue. So that's the measurement problem. And I would argue that in addition to building ND imaging systems that improve throughput and balance the trade-offs that we are all familiar with, it's also important to consider what computation can do for us and design all of these pieces together using deconvolution or inverse algorithms, or now foundation vision models. We can really relax some of the constraints that we face when we are working just with optical processing of information that we collect from the samples. So we do joint design of data acquisition schemes and inverse algorithms and foundation vision models, so that we can make this measurements at scale and with high accuracy and in a robust fashion. So that's the work of computational imaging. Now I'm going to talk about a couple of computational frameworks today. One is a generalist restoration and deconvolution method, which we call wave order. It's a wave optical framework that allows you to get quantitative estimates of material properties in both fluorescence and through scattering. It's adaptive. You can learn the parameters of the deconvolution from the data itself. So it is equivalent to shift variant blind deconvolution method. And we have developed this with biologists and to solve some specific biology problems with our colleagues. The work is driven by Talon Chandler, who is the computational imaging scientist, and Shri Vara, who is a research software engineer on the team. So I'm going to first start with some examples of data we can collect. So we are looking at here quantitative phase images of A549 cells that have been perturbed with a pool of perturbations. And you can see that from this data, we can see subcellular architecture. Also the phenotypes of cells. There are large cells, there are small cells, cells with vacuoles, cells with different structure of nucleoli, mitochondria. So there's a lot of information packed in this data. So I like to say that we're packing biology in every pixel through this strategy. And I'll talk about how this results. How do we get this images? And this is another application of wave order where we are really looking at three-dimensional structure. Here we are looking at an organ called a neuromast in zebrafish. This is the phase image, and at the bottom we have fluorescence image. Both of them have been restored or deconvolved through this framework, through wave order. And we are now working towards scaling the algorithms and its ability to deconvolve sample-dependent aberrations to the whole zebrafish. So that's a work in progress. So we have some proofs of concept, but it's not in production mode yet. So just to give you a bit of a background around how QPI, this flavor of QPI, we are really building on top of a very, very classical idea of phase from defocus that is still the primary method that's used in electron microscopy. But the idea is that if you have a refractive object like lipid droplets, if you look at them at different depths, at different focal planes, the contrast will change. A lipid droplet will appear dark relative to the background on one side of the focus, transparent at the focus, and bright on the other side of the focus. And this has to do with how the light is scattered and how it interferes with unscattered light and contrast variations. So it has to do with the phase of scattered and unscattered light and how that changes over propagation. What we want to do is to get a cleaner representation of the material property, the refractive index. We want data that says that lipid droplets are denser. than the surrounding cytoplasm, and nucleoplasm is less dense than most of the cytoplasm, and we want those measurements, and we don't want images that are in arbitrary units. We want images that have physical units. So all of us are familiar with deconvolution, fluorescence deconvolution, where you take a 3D stack, you have a representation of the microscope as a filter, and that this is the optical transfer function. And really, deconvolution is about doing inverse filter while being robust to noise. And you can formulate this problem of QPI in the same language. We are really doing inverse filtering, but the filter looks a little different. The filter has a slightly different support, as you can see from the shape, and also has different complex values because of to really model this contrast reversal that we just saw. When we have a fluorescent bead and you go above and below focus, the contrast changes symmetrically. When we have a refractive object and we go through the focus, the contrast flips. So we model that through this OTF that has change in the phase as you go on the either side of the focus. So once you do that modeling, and if the model is accurate, and that's the key piece, that the model needs to be accurate to your acquisition, that specific field of view, then you can really remove the microscope from the picture and really get to the biological sample. That has been the challenge for deconvolution, and that's what WaveOrder solves. Here is an example. Even after we do deconvolution with an initial guess for what the transfer function looks like, you sometimes get really bad results because the OTF is not properly calibrated. But and you can barely see here nucleus, nucleoli, organelle, etc. But if you do auto tuning and calibration from the data itself, you can get results like this. So here now we can see, of course, the structures, but with much sharper definition. We can see vacuoles. In addition, we can see membrane boundaries. We can see now mitochondria become a lot more visible. So how does this work? In this specific scheme, we are doing 2D phase from defocus. I'm going to frame this as a computational graph. And the first node, the central node in the graph, is the deconvolution node, where we are modeling the relationship between the object, which is the quantity we want to reconstruct, the quantity f here, and the data through the transfer functions. So let's say we have a few 7-slice Z slices. We can model that as bank of 7 different filters, and that bank really filters the spectrum of the object, and that results in the images. We have the actual images. So the optimization problem that we are solving here is estimate the object f that best describes the data D, assuming the filtering operations specified by the imaging system, by the any of the objective, any of the illumination and wavelength. And we have some regularization because this problem is ill-posed. So we have some regularization, that's standard Tikhonov regularization. This would work well if H were calibrated properly. Now, calibrating H properly is the tricky business. And what we have realized is that instead of trying to learn all of the parameters of the filter itself, it's better to constrain the parameters of the filter through some knobs, a few handful of parameters, such as the parameters of the pupil, the illumination angle, etc., and defocus. So here what I'm showing you is an extension of the graph. On the left side, we now add a computation that takes you from some handful of parameters to the transfer functions. And with that, you do this inversion. And as you sweep the parameters, you can see that the estimated object changes. Now, what we need to do is to tweak this parameter, to optimize this parameter, so that the estimated object is good. It's represents biology, and at the same time, we get an optimized version of the transfer function. So this is a reformulation of the blind deconvolution problem where you need to estimate the object as well as the optical transfer function. And we find that some simple losses, if you add loss on top of the reconstruction, and some simple loss here, in this case, you're just maximizing contrast in the middle of the passband, you can optimize these parameters. And you can do that at scale. So now we have implemented all of this in PyTorch, and it's now operational. We work with GPUs, and with this strategy, you can tile up your large data set, like the optical pool screen, learn the parameters, learn the reconstruction parameters for each tile, do the reconstruction and blend. That's how we get this large-scale label-free imaging that is still high resolution. It doesn't suffer from things like meniscus effect that we are all familiar with, or mismatch in the focal plane as you do the acquisition. And Gail, who we just heard about in Alfred's talk, he has been really having a time with this data and finding very interesting phenotypes, looking through all of this data and discovering new types of responses to perturbations. It's been really proving very valuable for optical pool screens. Great. So now we are extending this idea, this strategy of auto-calibrated reconstruction to whole zebrafish in collaboration with Loic. These are some of the early results where we are looking at 3D tissue architecture along with two fluorescent channels. One is a nuclear marker, and the other is a marker of differentiation of skeletal muscle. I'll just show this cartoon to illustrate that this idea applies not just to phase imaging, but also to polarization imaging. WaveOrder is a vectorial wave optical framework, not just scalar framework. This animation is done by my son, who was inspired by Lucky to take up Blender. So what we are looking at here is a very simple geometry where we introduce two components in the light path. One is a polarizer, programmable polarizer, and an analyzer. And with that, you can collect polarization resolved data, and from that, you can do polarization imaging. In interest of time, I'm going to skip over, not show the data that we have collected with this system. WaveOrder is pre-printed. There is actively developing GitHub repository. All the work is in open. There's a Hugging Face demo if you want to go give it a try with some of the data we have collected, and also a notebook if you want to plug in some of your transmission Z stacks and see how you can do QPI with this. How am I doing on time? Do I have a few more minutes?
Yes, like about a minute and a half or so. Yes.
Great.
Go
Thank
ahead.
you.
Yeah.
Thank you. Thank you. So this is great, right? So now we can visualize a lot of biology. We can see a lot of cells and their phenotypes. But to really get to that stat of reading out molecular responses or organelle responses, we need computer vision on top of this data. That's also as robust. So WaveOrder is robust because it's constrained by physics. It's a learned model, but it's constrained by physics. So it is useful for many different cell types and tissue types and many different types of microscope configurations. And that's classically not been the case with computer vision models. They work with specific distributions of data and fail when you go out of distribution. And we have been trying to solve the problem. I'm not going to don't have the time to talk about how we did that, but I'm going to show some results to illustrate the features of this class of library of models, which we call Cytoland. So the goal with this models is to allow you to virtually stain landmarks in the cells and do it robustly across cell types and across different instruments, different types of input label-free data. Could be phase image, could be Zernike phase image, could be bright field image. And depending on how much information you have in the input, you would get different quality of results, but the model should work, same checkpoint should work with different types of label-free inputs. So that's the goal. And the motivation, the biological motivation, is to free up the spectrum to look at additional information. We shouldn't be using up 2 channels of fluorescence just to look at landmarks because those structures are visible in one label-free channel. And more is visible, like mitochondria and in specific cell type. So we want to free up this fluorescence channels, and we want to do this in cells and different types of cells and tissues. This work was driven by Zivan, who is now a PhD student in Germany, and now by Ed and Alex, who are at the Biohub. These are some of the things you can do with robust virtual staining. You can do single cell tracking. So we are looking at here a slice of a 3D phase image, and then these are virtually stained nuclei and membrane. These are not experimental stains. These are purely virtual stain. That's why they look denoised. And from that, by using Loic's Ultract, we are able to do single cell tracking at scale and robustly. We have also found that we can use these types of models to do single cell tracking in the organs of the zebrafish, and we are trying to scale these models to the whole zebrafish. All of this work is summarized in a paper that came out last year. Again, we have a GitHub repository and an interactive demo. And if you're familiar with agents, the code is written well enough, documented well enough that Claude or Codex, whichever agents you're using, you should be able to use these tools in your own workflow. And so I'd like to close by thanking my team. We are a highly collaborative, interdisciplinary team, and many collaborators at the Biohub.
Great. Thank you, Shalin, for your presentation. And thank you for all the speakers. It's amazing presentation given by all. And now we will start our Q&A session. Anyone with questions, please feel free to unmute yourself and ask the question, or you can put it in the chat. Anybody wants to go first?
I have a quick question for Kevin. You said that you're using Zemax as part of your workflow. I was wondering, so you need a Zemax license, right, to do that? Have you thought about that?
Yeah, we've thought about it, but haven't done much about it. So it seems like there's a lot more competition in the market now, including some more open simulation systems, and I think that's probably worth a shot now to explore. Now that John's moving on into industry, that the next person should maybe look into some more open-based things that don't require that license.
Thanks.
I guess while we're waiting, I just comment that I'd really love to see it go that way. There is a lot of open source ray tracers now, and I think some of them are potentially quite powerful. There's just this little hurdle of when you go to a manufacturer's website and they offer you up like the Zemax black box or file, and then it's kind of awkward, right? Because if you don't have Zemax, you can't necessarily model something. So it'd be nice if that could flip over to, and we also offer, I don't know, something else.
Yeah, this discussion happened also in the last session where we were talking about maybe trying to gang together and build some sort of open source alternative. So it does seem to be a recurring theme.
I had a quick question for Kevin, if that's allowed. Okay, great. Thanks, Kevin, for the great talk. I realize this work's quite recent. I don't know if you've got any sort of guinea pig labs lined up to provide some feedback to see what it would be like to try and build one of these systems without any other input other than your documentation.
Yeah, so I've actually had 2 Chinese labs reach out, and then I was unfortunately advised that I would be subjected to FBI monitoring if I then worked with them. So I advised them to refer to our GitHub pages as much as they can, and I think that they're making progress. And we have Andrew Weems' lab down at University of Texas in Austin has been doing it. and a lab at University of Oklahoma were helping with do it. But we have yet to have someone that's just purely non-optics build one.
Okay. Have you got any sort of early feedback that you can share?
Early feedback. I have not gotten too. I think there was like one. Yeah, some sort of. The approach that we have is if we do get a question, we then look at our documentation, see if we addressed it at some point, and then if not, we try to update it. I think that there was some ambiguity when someone's looking at like the focus after a cylindrical lens, and you could look at it's focused in one dimension and it's collimated in the other, and they were confused by, I thought the flat side was supposed to be towards the focus, and so that's about the limit of the feedback that I've gotten. On the software side, it's a little bit easier, and I think the feedback I've gotten have been frustrations with like the initialization of the configuration, which is a frustration I have myself. So creating better helpers that then kind of allow people to launch a system from scratch is what we've done there. And that was inspired by feedback.
Okay, great. Thank you.
Yeah.
Reto, you have a question?
For Shalin, I hope I can phrase it in a short way. Can you hear me, by the way?
Yes.
Yes.
Okay, cool. The phase imaging, especially quantitative phase imaging, if I see it the right way, there has been a lot of work on, say, Born approximation, other approximation with coherent light, like nanolife and HomoCube. And lately I learned Shefta-Dip Chowdhury at
Shweta-Dip.
He solves even a layer by layer inverse problem just from a hologram without even doing much about it, and somehow this converges. So there is that field with coherent light and inline or off-axis holography or holotomography. And then there's also Michael Schriebach with the multidirectional DIC as something in the incoherent realm that can use the full condenser. Could you maybe try to help me where your technology fits in? In that space?
It's closer to Michael's technique. So it is partially coherent, just like FPM or DIC methods. And there are several variants of this qualitative phase imaging that are becoming quantitative. This phase from defocus that was qualitative. Now we have made it quantitative, and it really scales well. There's differential phase contrast that was qualitative. There's quantitative DPC. There is a camera from PhasX in France, which we could make more quantitative. So yeah, this specific class of methods, the whole wave order library, supports incoherent and partially coherent quantitative imaging, incoherent fluorescence, partially coherent transmission, and also with polarization diversity, if that matters for fluorescence or transmission. And what we have realized on that mathematical note, what we have realized is that there is a unified representation. So if you dig into the preprint, you'll find that there's a beautiful connection with spherical harmonics. You can represent the properties of the material, the scattering potential, and the fluorophore distribution with in terms of the spherical harmonic basis. And that's what allows us to represent this large collection of methods with one forward model and invert with the same algorithm, if that connection helps.
But if I may quickly, on a practical note, is a selling argument that this can be implemented on, say, existing bright field microscope, whereas a holotomography setup is more involved. Is that maybe a practical
Yes,
consequence?
from the usage point of view, that's exactly right. So we have multiplexed these ideas with confocal, with snout scope, with wide field, with ISIM. And whereas if you were to take coherent approach, you would have to really retrofit fluorescence to this method, to whatever method, coherent method you choose, holography-based method you choose.
Thank you. I leave it at that, even though I could probably chat with you for another hour.
Yeah, we can chat another time. Yeah.
And then Spencer's question already answered by Gail in the chat, so that's good. Any other questions?
I have a really quick one for Kevin. I really like the Altair-dvOPM. I just think it's super elegant because you've chopped out so many optics. I'm just wondering with those big photography lenses, how do they compare on transmission efficiency to, I don't know, an objective or on some absolute value? Just curious.
Yeah, that's a very good point. That's something we should measure.
Oh, okay, because they don't give it, right? Yeah.
Yeah, and so we'll send a beam through and measure that because
be cool.
Yeah, having the fewest elements as possible is always best for photon efficiency, and I'm curious how that actually works out there.
I imagine they're pretty good. I wonder about coatings and things, and maybe you could get one and recode it and get even more. Not that it's necessarily worth it, but yeah.
It sounds like a luxury. But yeah, no, that's a good idea. I'll report back.
Yeah. The selling point was that these fast photographic lenses are comparatively inexpensive. For personal use, they're expensive, but for microscopy use, they're surprisingly cheap.
Shalin, yes.
Yeah, I had a question for Kevin, Gail, and Alfred, all three of you, around how do you think about restoration, deconvolution? Do you write code yourself? Are there specific algorithms that you found to be very useful for the type of fluorescence methods you are working with? And Gail, you talked about OpenFPM. I'm really curious. What sort of algorithm works well for estimating the phase from the FPM data?
I can.
Yes, please. Yeah.
OpenFPM first. We are using the algorithms of others for the reconstruction because they already exist. Why should we reinvent the wheel? That would be ridiculous. To answer your question about deconvolution, honestly, it depends on the question that we have. If it is, we have a data set that's well known and well understood, and a lot of the proprietary software methods work. We have a copy of Huygens, we use it, it's fine. When it's something a little bit extraordinary, we will try the proprietary methods, and when they fail, we try and write our own.
Yeah.
So anything that anybody does that can stop us having to do that is most welcome.
Yeah, I would say out of laziness, I prefer not to do any sort of deconvolution whatsoever. And if I can present the data as is, then that's the nicest, cleanest format. There's no parameter tuning or hacking that needs to be done to produce, an output that may vary between labs. And so I think that's always made me uneasy. Nonetheless, you obviously have to do some deconvolution sometimes to get the glossy image, which can be favorable in some manuscript context. And there we have kind of a blind deconvolution followed by Richardson-Lucy deconvolution method. So the blind estimates the PSF, and then that's then applied to the Richardson-Lucy that Bo Ray and Reto's lab kind of coded up in MATLAB. And I'm also quite happy with PetaKit 5D and their Python wrapper, which I use because it's performant and fast, and I don't touch a parameter.
Alfred, what's your toolkit for deconvolution?
Yeah, so the short answer is I don't use it. Yeah.
Okay. Yeah, it sounds like it's totally optional for fluorescence when you have really good results, but it is really critical when you do phase imaging.
Shalin, just a quick question. I don't use deconvolution. How fast is it Like, is it computationally very expensive or?
It is expensive. But now these days we all have GPUs, and with GPU, and it is relatively fast. It's certainly faster than CellPose, for example, per field of view, to do one step or a few steps of iteration. So, we've implemented this in PyTorch. We are still working through scaling this to very large data sets, but on relatively small data sets, it works on a laptop. It works on a standard lab computer. If you have a GPU, it's faster than running CellPose for segmentation.
Great, thanks.
Any questions from the audience? If there's no more questions, maybe we can wrap up our talk early today by a couple minutes. Again, this whole session was recorded and will be uploaded on BINA's YouTube site. And our next series will be in the fall, so keep an eye out for that. And if there's nothing else, we can end the talk, end the meeting here. Thank you, everybody, for joining today. And once more, thank you for all the speakers. Great presentation today.
Thank you.
Thank you. Bye.
Great.
Thank you.
Thanks, everyone. That was amazing.